Yeah, okay, we'll start the show then. Do you want to pop on the next slide? Thanks. Welcome everybody. We have tonight a talk by Dr. Rosa Santomartino, who's going to talk to us about astrobiology, and Astro-Rock, sorry. Ahead of that, just a quick reminder really, the website for the Astronomical Society, the address is on the top line there. You can find out what's happening within the society. You can find our old meeting notes there, the videos there, etc. On Facebook, you can follow us there, or on Twitter. And indeed, our own YouTube channel will give all the talks that have been recorded since lockdown. So find us, follow us with pleasure. Coming up, there's been a slight change to the agenda. Mark's introduction to astroimaging part 5 has gone back to Friday the 24th. And we have Bruce plugging that gap on the 15th of July with a story about Thomas Henderson, the first Astronomer Royal for Scotland. So an exciting programme that gets better every time I look at it. So with no more ado, I'm going to hand you over to Rosa, who's going to talk to us tonight. Would you like to share your slides then, Rosa? Yes, no?
Would you like to unmute yourself?
Yes, probably. I was talking with. Oh, sorry. I don't know why it doesn't start from the beginning. It's a lot of slides, I'm afraid, but it's just because I don't like animations, so I just copy the same slide. Okay. Okay. Can you see it correctly?
We've got the presenter's view.
Okay, let me close it. Apologies. Let me try again. It was better before. Okay. What do you see now?
That's excellent.
Okay, amazing. Okay, so thank you very much for inviting me, first of all. I'm very excited of being here, and thank you for giving me the opportunity to talk about the BioRock project that is a space experiment project that was launched on the International Space Station almost 1 year ago in July 2019, and I was lucky enough to lead it together with my supervisor, Professor Charles Coquel from the University of Edinburgh, and of course together with the whole BioRock team that, as you will see, it's quite a large team. First of all, I want to go on with the slide. Okay, so what we are going to talk about today. First of all, why did I wrote "we" in the title? So what do I mean when I say "we"? In other words, what is a microorganisms? Which are the roles of microorganisms on Earth? And in particular, I'm going to talk about one role that I'm very interested in about, that is biomining. And then which are the possible roles of microorganisms on Earth and beyond? And so again, biomining, but this time biomining in space. Then I'm going to tell you something about the BioRock experiment itself and something about what to expect from the future of this. So first things first, what is a microorganisms? I wrote "we" in the beginning in my title. So why did I put that? Let's get to that. This is the simplified version of the evolutionary tree of life that we biologists use normally in biology to describe the whole diversity of the living organisms that are present on Earth and that we know. As you can see, these are extremely diverse, and they include organisms such as bacteria, protists, fungi, animals, in which there is also humans, and then plants and also archaea. They all derive from a first organisms, that is the so-called LUCA, or last unknown common ancestor. But when we talk about microorganisms, what we are referring to is in particular these organisms here. There are bacteria, archaea, protists, and fungi, in particular unicellular fungi. Indeed, microorganisms are a wide variety of different types of organisms. For example, unicellular algae, archaea. There are some very interesting organisms that in the beginning, when they first discovered them, they thought that they were bacteria, but then they discovered that they were actually quite different from the biochemical point of view from bacteria. And then we have bacteria, of course, that are the most famous one, maybe. Fungi, in this case, I'm referring to unicellular fungi. Viruses, and that are quite famous, unfortunately, in these times, and protozoan, that are the largest type of microorganisms that are known. Why microorganisms? Microorganisms, because these organisms are very, very small and tiny. In fact, they are in the order of microns. Microns is 1 micrometers, means 1 micrometers, and 1 micrometers is actually 0.001 millimeters, or 0.000001 meters. So, for example, if this is a needle, as you can see, this is a needle. A needle is more or less 1 millimeter wide. This is quite a wide needle, to be honest. And if you take a whole millimeters and you take an organism that is quite known and studied in my field, there is astrobiology, such as tardigrade. Tardigrade is a very tiny organisms, but it's still in the orders of millimeters. In fact, as you can see, it's big, it's half of a millimeter. While if you take an organism such a cell like a human egg cell, that is one of the biggest cells that are known, it is in the order of, still in the orders of millimeters, although being quite small because it's 0.1 millimeters. If we take the biggest microorganism that is known, there is a parametrium, there is a type of protozoan. So as I said before, one of the biggest microorganisms known, it is more or less as big as a human cell, egg cell. Now, if we go further and we take 1/10 of millimeter and we enlarge it, we have 0.1 millimeters or 100 micrometers. At this size, the parametrium that we observed before is actually as big as this. And if we go to the next microorganisms in terms of size, we have cyanobacteria. This is an example of cyanobacteria that is still quite big in confront to the smallest microorganisms because it's more or less 50 micrometers. If we go further, we go to unicellular fungi, for example, baking yeast or mold. These organisms is in the order of 10 micrometers. Let's go further and let's take 1/10 of this, that is 10 micrometers, of course. In this order of magnitude, the fungi, unicellular organisms, is as big, while a bacteria, finally we get to bacterium. Here there is an example of the most common bacteria that is known, Escherichia coli, maybe you heard about it. It is so small that compared to the fungi, it is only this big, and it is in the order of 2 micrometers, more or less. Then we go further, and there are viruses. For example, smallpox virus, that is one of the biggest viruses that is known, is still in the orders of less than 1 micrometers, basically. And then COVID, for example, is quite small. It is 0.06, 0.1 micrometers. So this is just to answer the question, why micro, why we? Because when we talk about microorganisms, we are really talking about very, very tiny and small organisms. And as we saw before, they are very diverse. But despite that, there are some characteristics that are in common between all microorganisms that are, First, they are all single-cell organisms, or even less. For instance, viruses are not cells, they are less than cells. And the second characteristic that is common to all the microorganisms is that they are everywhere on Earth's biosphere. When I mean that they are everywhere on Earth's biosphere, I literally mean everywhere. For example, they have been found on soil, they have been found on pipes. For instance, in this picture here, you can see that this pipe was damaged by frost, but also by bacteria. They have been found in very, very hot environments, such as hydrothermal vents, very dry environments, such as deserts, in clouds, for instance, very salty environments, such, for example, the Dead Sea, that is not so dead after all, because it contains such bacteria and archaeobacteria. Then very cold environments, such as Antarctica, and also animals, as for instance, us, human body. And why it is like that, why they are everywhere and they are so successful? It is because they are very simple. So the fact that they are extremely simple makes them also more adaptable to all the different environments that are available on Earth. And this is just a curiosity for you. It has been estimated that for each human cell that composes the human body, there is one bacterial or microbial cell, meaning that there is a ratio of 1-to-1 from human cells and bacterial cells. This can be scary if we think about microorganisms as a nasty thing, but this is actually quite very, very good, because these bacteria and these microorganisms do a lot of very good things for us, and in particular they are extremely good for keeping some equilibrium that makes us healthy. So as I said, the majority of time when we think about microorganisms, we think about something nasty, but for example diseases, infection, and something like that. But the reality is that most of the time they are silent, necessary companions of our life. And I can make you tons of examples, but I will try to summarize them like that. I want to show you and to demonstrate you that what I'm saying is true by listing some roles of microorganisms do on Earth for us. For instance, they are extremely useful and necessary for food production. Food production, for example, bread, of course, alcoholic beverages, but not only, cheese, pizza, and a lot of other food types. But they are also extremely useful in bioindustry and in biotechnology. For instance, they are studying now how to produce biofuels from microorganisms, so some kind of green energy. Also, they are extremely important for the production of oxygen on Earth. In fact, not only plants produce oxygen, but also some kind of bacteria that do photosynthesis, such as cyanobacteria. And indeed, in early Earth, the oxygen percentage in the Earth atmosphere was not as high as the atmosphere that we have now. So we have the percentage of atmosphere that allowed the development of developed life, such as humans, for instance, because of the actions of some cyanobacteria that produced at a certain point what is called the great oxidation event, that produced a lot of oxygen in the atmosphere, and allowing that for the development of human life, for instance. Then, from the biotechnological point of view, microorganisms are also extremely useful for the production of compounds, such as, for example, cotton and wool. You know that this has been produced by a plant, and this is produced, of course, by animals. But actually, some biotechnologies are now able to produce these type of polymers from bacteria. And I also like to mention this one. This is biocomposite. Biocomposite is a real type of composite that is self-healing. So basically, it is a bioconcrete, basically. It is a concrete that can self-heal when there is a crack, for instance. And it is also thanks to microorganisms. Another way to be useful for us is the production of biomedicines. For instance, maybe you may be aware of the fact that the first antibiotics came from a mold. A mold is a unicellular fungi, so a microorganisms, and from this, a lot of other antibiotics have been discovered starting from microorganisms. But also, again, in biotechnology, there are a lot of productions of medicines that are possible thanks to bacteria, such as, for instance, insulin. And then they are also extremely useful in recycling, for instance, recycling of waste, also plastic. In fact, some microorganisms that are able to degrade PET and plastics in general have been recently discovered and bioengineered, and also electric waste. They are also useful for environmental matters, for instance, bioremediation. Here you can see some pollutant environment, and maybe the image is a little small, but you can see that this researcher here, well, I'm not sure they are researchers, but these men here are actually spreading some bacterial moisture that will degrade the toxic pollutant that have been spread on this beach. Another very important role of microorganisms is in soil production. In fact, without the effect of bacteria and microorganisms in general, soil will be just an inert abiotic matter. Thanks to the enrichment of organic materials mediated by microorganisms, soil actually becomes fertile. And last but not least, of course, biomining. Biomining is a very interesting process that I'm going to tell you more in details in a few slides. But one thing that I want to underline here is that, in particular, these last processes here that I show you are linked by similar chemical reactions. The chemical reaction that I'm talking about are the reactions of bioleaching. So basically, the bioleaching is a complex biochemical processes that is thanks to the fact that some microorganisms use chemical elements such as sulfur and iron as source of energy. These sources of energy are of course present in rocks and in soils. So what they do is that these bacteria try to get these nutrients like sulfur and iron and these source of energy from the rocks, and by doing that they attack the rock or the substrate in general, and doing that they weather the rock, they produce soil, and they also free useful elements that can be therefore be useful for us. And these elements are sometimes elements that cannot be extracted in other ways than by bioleaching. One of the application of bioleaching, the industrial application of bioleaching, is actually biomining. The definition of biomining by the AGI definition, by AGI, is the process of using microorganisms to extract metals of economic interest from rock ores or mine waste. And also for bioremediation, that is that technique that I told you before. On Earth, they are extremely, it is wide used, for instance, for the extraction of copper, gold, nickel, cobalt, and other elements, such, for instance, rare earth elements that I'm going to tell you more later. And there are several advantages in respect to the classic mining techniques. In fact, sometimes they can be complementary to traditional mining technique because it improves the rates of metal recovery. For instance, there are a lot of waste dumps and heaps coming from traditional techniques. These mine waste are basically material that in which the useful elements cannot be extracted anymore by traditional techniques. In this case, biomining can actually really help because bacteria can, as I say, can help make these elements available for further extraction. It is also quite an economic technique because of the fact that if you think about it, the only thing that you need is the substrate that can be mine waste, a horse, dumps, heaps, or something like that, and some water and some nutrients to make the bacteria happy. But that's everything that you need. It's very, very economic. And it's also environmental friendly because some of the chemical wastes that are produced by technical mining, traditional mining technique are actually not produced anymore by biomining. So it is actually quite advantageous and widely used technique. But as I promised, I'm going to show you not only how microorganisms are useful on Earth, but also in outer space. In fact, as we move into space, we can harness microbes to make our life easier and improve the success of space settlements. So not only which are the roles of microorganisms on Earth, but also beyond Earth. And let's see which one these roles could be. For instance, first of all, they could be useful to generate oxygen. We saw that they were extremely useful to generate oxygen during the Great Oxidation event on early Earth. If we move to another planet, we know that the oxygen percentage in the atmosphere is going to be different. So microorganisms can actually be extremely useful for us to produce the right amount of oxygen for us to breathe freely. But also to generate food, because as we can imagine, we are not going to have a lot of source of food on an extraterrestrial environment. So microorganisms can help with that as well. But they can also produce soil that is strictly related to this, of course, because starting from extraterrestrial regoliths that are suboptimal for growth, we can actually, microorganisms can actually add those organisms, those organic material and compounds that can make the soil, extraterrestrial soil, finally fertile and improve the carbon content. They will be also extremely useful to recycle waste. We saw that this was extremely important on Earth, but in a closed environment such as a space station or an extraterrestrial settlement, this becomes I would say this becomes essential because we are going to recycle as much as possible. In fact, if you think about it, it is already a thing on the International Space Station. For instance, they continuously recycle their water because, of course, International Space Station is an enclosed environment. This is going to be extremely important. Last but not least, in one day in which we are going to be completely settled on an extraterrestrial environment, we also need at some point, unfortunately, to produce drugs. And as we saw, as microorganisms can do this on Earth, they will be also able to help us on an extraterrestrial environment. All these possible applications have all a final main scope, actually two final main scopes. First, allow and develop life support systems. And when I mention that, I like to mention also this very interesting project that I really like, that is the Melissa project. Melissa stands for Microbiological Life Support System Alternative, and it is based and expired on terrestrial ecosystems. And basically, it is a research project that has the aim to develop an advanced support system based on different microbial, actually not necessarily, a loop of 4 compartments. Each compartment basically with a specific organisms and with a specific task. For instance, there is going to be a compartment for atmosphere regeneration, for respiration, another for water recycling, another for waste treatment, another for generation of food, and so on. But what is interesting about this project is that every compartment will produce something that is useful for the next compartment, but there is going to be a waste for the first compartment, and consequently they are going to use as carbon source or an energy source, the waste of the previous compartment. Interestingly, also, there is a fifth compartment that is actually present, that is the human crew. In this case, you can see the astronaut. The second approach that all these roles of microorganisms is going to help is the in situ resource utilization. The in situ resource utilization is an approach that can be summarized thinking about the fact that someday when we will go on Mars or Moon or another space, we are going to need to be independent. We can about relying on constant resupply from Earth, but this is going to be extremely ineffective and extremely expensive, especially if you think about the fact that transport 1 kilogram from Earth to the Moon cost of each material cost $100,000. So this is going to be extremely ineffective, and we need something that is way more sustainable. One example would be biomining in space. Of course. In fact, as I was saying, as humans plan expedition to places such as Moon or Mars, biomining offers a way to obtain the needed materials on other planetary bodies rather than bringing them from Earth. So a way to go to that approach of in situ resource utilization. When we talk about biomining in space, we are talking about a field that has an increasingly interest in the community, in the space science community, basically. The planetary body that we are interested in mine are, for instance, Moon, Mars, or asteroids. Moon and Mars are rich in basalt, and basalt is also present on Earth, and it's rich in elements such as a lot of rare earth elements and copper, nickel, vanadium, other trace elements, and so on. So we are interested in trying to take these elements from Moon or Mars for an extraterrestrial settlement or for the terrestrial use. And then there are asteroids. Asteroids are very, very rich in expensive and rare elements such as platinum group elements, volatile, silicates, nickel, irons, precious metals, gold, iridium, and so on. You can understand why people is getting more and more interested in trying to understand how to biomine in space. And as I was saying before, the interest about biomining in space is not only from the point of view of creating a space settlement, but also from the point of view of terrestrial industry. For instance, I like to show this image here from a project from Luis Zea research page from the University of Colorado. They have this interesting concept of imagining a future in which Earth is a zone that will be reserved for only living, and the heavy industries such as the mining and industries are going to be moved offward on asteroid, Moon, or for instance Mars. It is in this frame of thinking about biomining in space that the BioRock experiment started. Actually, the biomining experiment first started going back in 2018. In fact, it was in the frame of the ESA geology, the biology for space exploration and settlement topical team that Professor Coquel from the University of Edinburgh, that is also my supervisor, first proposed the BioRock experiment. However, the BioRock experiment, you may notice that being 2020 now, it took 12 years to be actually ready, 11 years actually to be ready for the real launch on the International Space Station. And this wouldn't have been possible without the excellent, complete BioRock team and the effort that you can see here, summarized here, and the excellent effort of various present and former researchers and students that were involved in the project. But which one was the motivation for BioRock? Of course, we wanted to test if mining of extraterrestrial materials was possible, and from both regolith, for in situ resource utilization, and also using biomining of rock and regolith life support systems. So we already talked about that. We know what we are talking about. And the main question of the BioRock was, however, okay, we know that biomining is possible on Earth because, as I showed you, this is being widely used and it's more and more used because of its advantages. But how do we know if it's going to be possible in space? For instance, in space, there are some conditions that are particular and are not present on Earth. One of these is the presence of low gravity, such as microgravity, or microgravity that is only 0.4 G in respect to Earth gravity that is 1 G. So the main question for BioRock was, how does low gravity influence the way in which microbes interact with rocks and mineral surfaces, and hence extract useful minerals and elements from the rocks? Now I'm going to share my audio here. You're okay. I'm sharing the computer sound because NASA, before the launch of BioRock on the International Space Station, produced this very nice video that explained perfectly the rationale and the concept of the BioRock experiment. So I would rather make them talk because I think it's going to be very, very clear. Oops. But I need. Okay.
The International Space Station will soon host some of the smallest miners in the universe, microbes. Microbes growing on the surface of rocks can gradually break them down and extract useful minerals and metals. This is a process called biomining. As we explore space, we are seeking to use biomining to turn rock and regolith into soil for growing plants and food. But before we can use this technique in planetary settlements, we first need to test it in space. On the space station, bioreactors will be placed inside a centrifuge where microbes will grow on rocks in microgravity and simulated Martian gravity. Investigators will examine how 3 types of microbes behave within pieces of basalt and evaluate how well the different microbes extract elements from the rocks. The findings will be compared to ground-based results. We hope to gain insights into how microbes interact with rocks in microgravity and how we might use them in our exploration of deep space.
Subscribe for more space.
Okay, I hope you heard the audio. And so this is the rationale for the BioRock experiment. So just in case the audio was not working, we wanted to test if, as I say, microgravity and Mars gravity will actually affect the way by which microorganisms interact with the rock and finally extract the element from the rock. So we needed to select the right organisms among the whole wide variety of biomining and bioleaching organisms, microorganisms. How can we choose that? We decided that the right microorganisms would have had at least 1 or better 2, 3, better all of these characteristics here. For instance, they needed to come from natural rocks or soil environments, because by this we knew, of course, that it would have been a good candidate to work on a rock surface, or it should have been already demonstrated capability of extracting elements from the rock. It should have been desiccation resistant because of how the samples were launched on the International Space Station, or they should have been used previously in a space experiment, because this helps a lot understanding the fact that they could survive and flourish on the space station, and also they can be easily studied in laboratory for our analysis, of course. The organisms that were selected in the end were these 3 organisms here. first Stringomonas dissecabilis that were provided by our laboratory, so here University of Edinburgh. Bacillus subtilis, there is a very is widely studied on the International Space Station. It's very resistant to a lot of harsh conditions and was provided by the German Aerospace Center, DLR from Germany, and Cupriavitus medallidurans that is has been discovered to be useful in bioremediation, basically. And this was provided by the Belgian center SEK-CEN. Once selected the organisms, we prepared the samples for the launch. So what we did, we have taken the 3 organisms and we inoculated them. So we grow them in a normal liquid culture. We inoculated them on basalt slides. Why did we choose basalt? You may remember that when I mentioned which rocks and which elements are present on Mars, I mentioned that they are rich in basalt. So we chose basalt because we thought that it would have been a good analog to understand if these organisms were able to extract useful elements from this planetary body. You may notice here that there was also non-biological control. The non-biological control was represented by basalt slide without any bacteria on them. And this is essential because when we went to analyze the samples and we needed to understand if the organisms actually did extract some element from the rock, we needed this control to understand if the elements found in the samples were due to the action of the microorganisms or if they were just leached by chemical processes that did not involve the microorganisms. Once prepared these samples here, they were inserted into this very nice hardware that is called the Biomining reactor or Amar that was specifically developed for the BioRock experiment. Here you can see a biomining reactor, an hardware in which 2 basalt slides were inserted, so 2 samples, and here you can see an empty one in which the basalt slides are not present. And moreover, here you can see an experimental an hardware in its final look, because here it's into its experimental container. Here again, this is a bigger picture of the whole hardware, and here is a bunch of hardware that were prepared for the experiment, the space experiment. After this, what can we show? I'm going to show you some nice pictures of us. This is me, of course. This is Professor Charles Coquel, and this is Annemiek Wagen, a PhD student in our laboratories that helped us for the integration of BioRock at the Kennedy Space Center. The NASA Kennedy Space Center. Here you can see us very happy because we finally finished integrating all the hardwares. And here you can see Charles delivering the finalized hardware ready for the launch, the International Space Station processing facility. And this is us. This is just a little part of the BioRock team that were present at the Kennedy Space Center, enjoying some tour of the NASA Center. Then finally, the experiment, the samples were launched on the SpaceX Commercial Resupply Service 18 here. Here is me on the launch side. This is a way better picture of the launch. And here you can see the Dragon capsule when it reached the International Space Station. And here you can see the astronaut, Luca Parmitano, that was the astronaut that took care of the BioRock experiment on board the International Space Station, that is installing an hardware into the cubic incubator on the International Space Station. Once they were installed in the incubator on the International Space Station, a whole set of samples were subjected. There were 4 complete set of samples, and each one was subjected to a different gravity condition. The first here was microgravity. There is a gravity you may be you may already know what microgravity is, but basically it is a very, very low gravity that is close to zero, but it's not actually zero, and it's present on the International Space Station. Then another set of samples were subjected to simulated Martian gravity. Another was subjected to simulated terrestrial gravity, and a fourth set of samples were actually not launched on the International Space Station, but it stayed on Earth and was included in what we call the ground control experiment that was actually essential to understand which elements were actually due to the gravity condition and not due to other conditions. Here you can see, for instance, the astronaut Luca Parmitano that is performing a post-growth image acquisition on board the International Space Station. So what he did, basically, he has taken the picture just after the start of the experiment and just after the end of the experiment. This set of pictures is what we call the photo session. And despite being just a set of pictures, it turns out to be very informative for us, because here you can see a whole bunch of pictures that were taken after the completion of the experiment. And from this, we saw that some features that were not present anymore when we retrieved the samples on Earth were actually present when the samples were still on the International Space Station. For instance, here, the yellow arrow shows these bacterial aggregates that, as I said, were not present anymore when we retrieved the samples. And also we were able to see with this blue, indicated by these blue arrows, that very tiny bubbles, air bubbles were present in microgravity. And then these tiny bubbles became bigger and bigger the higher was the gravity regime. Finally, the BioRock experiment ended, the growth of the samples were completed, and we, you can see here, Charles and I, retrieving the samples, very happy and relieved that everything was going fine, went fine. So in the end, what did we discover? Is there something interesting? Which are the results that we can show here? Of course, we had very, very good results from BioRock, and I'm very happy to show you these. The first result that we had is that actually Mars gravity and microgravity did not affect at all the bacterial growth after 21 days. This is a complex plot, but basically what it says is that the gravity condition, so Mars gravity, microgravity, Mars gravity, Earth gravity, and ground control did not affect at all the bacterial growth after 21 days of growth for each bacterial sample. What does it mean? It means that, first of all, I need to underline these. When I say after 21 days, this is a very important point that I have to underline, because of how the BioRock experiment was designed, we were not able to understand the bacterial culture during the experiment, but we were able to understand what happens in the end of the experiment, so after the 21 days of growth. This means that the fact that we saw no difference in the final cell concentration means that doesn't matter how the bacterial growth went during the 21 days of growth. It doesn't matter, because in the end, after the 21 days, the bacterial culture all reached the same cell concentration, regardless of the gravity condition. And this is a very good news from the point of view of biotechnological applications, because this means that biotechnological application on Mars, Moon, and other celestial bodies will be possible. It is very likely they will be possible, because gravity condition won't affect this. So this is the first result that we had. So the first result was low gravity will not limit biotech applications in space. Very good, right? But what about biomining? Well, we also have results regarding biomining. And to tell you something about what we discovered from the point of view of space biomining, I have to introduce you very shortly about rare earth elements. They are a group of elements that you can see here on the chemical periodic table that are characterized by similar properties. And despite the name, they are actually quite abundant on Earth. And the reason why they are called rare elements is just because when they were discovered on the 18th century, they seemed quite scarce. But it became more and more evident that they are actually quite present on Earth and abundant. However, they are not very easy to extract. That's why they are very important. And another reason why they are extremely important is that there is enormous impact of rare elements on our daily lives, because they are extremely present in particular on electronic devices. We use them every day, but we are not aware of them. Where are they present? Well, they are present on Earth. As I say, they are quite abundant, despite not necessarily easy to extract. But they are also present on Moon. For instance, they are present in a particular rock type that is called AREEP, that stands for potassium, rare earth elements, and phosphorus. But they are also present on Mars. Actually, they have not been discovered on Mars by rovers on Mars yet, but they have been found on Martian asteroids that have been found on Earth. So we have evidence that they are present on Mars, and also they are present on asteroids, of course. So we discovered some rare elements biomining from the BioRock experiment. Of course, we did. In fact, one of the most important results that I can show you here is that one of the 3 organisms, that is the Sphingomonas dissecabilis bacterium, was actually able to biomine rare earth element in space in quite a high extent. This is quite a complicated graph. I'm sorry about that. The analysis of the bioleaching and biomining results, it's very, very complicated, but this is the easiest, the simplest plot that I can show you. And it shows basically that Sphingomonas dissecabilis was able to biomine rare elements in all the 4 gravity conditions that we tested for BioRock. What does it mean? It means that first of all, as I say, that Sphingomonas is able to extract useful elements regardless of the gravity condition. And this has some interesting implication, because this indicates that space biomining is in principle achievable under a range of gravity conditions. So very good news from the point of view of space biomining. Again, so BioRock is actually provide the first demonstration of biomining in space. Last but not least, as I mentioned before, the BioRock experiment allowed the production of the development of this space biomining reactor called Amar. There is an interesting and very intelligent and smart device that was used for BioRock, but is actually will be actually very useful for further experiment. And in order to demonstrate that, I'm going to tell you now that we have a second biomining space experiment that is going to happen hopefully by the end of 2020. That is going to be called BioAsteroid. The rationale is very, very similar to BioRock, but this time we are going to biomine asteroids rather than terrestrial rock materials. And also in this case, we are going to use other organisms. First, the bacterium Sphingomonas dissecabilis, as it behaves very well for BioRock. But this time we are going to use also unicellular fungus, that is Penicillium simplicissimum, and also a combination of the 2, because if you think about it, when we think about a real natural environment, we don't see single microorganisms, but we actually see a community of different organisms. So we want to see what happens from the biomining point of view when we have a kind of small community. So what can we expect from the future of biomining, but of microorganisms in space in general? There is a lot yet to understand. For example, we still need to select the best microorganisms that can biomine or produce other useful tasks for human settlements. We saw that Sphingomonas dissecabilis was a good one, but there are of course others that will be good as well, and we just have to discover them. Then we also have to discover how they are going to be affected by the long duration space flight. So, okay, we performed the BioRock experiment for 21 days, but what happens when we have long-term space settlement? Are they going to be more efficient, less efficient? Are they going to be affected somehow? We have to discover that. We also have to develop the technologies. For instance, how to collect the asteroids. Here there is an image that if you Google biomining of asteroids, you will find content of different projects that will try to understand how to possibly collect the asteroids, biomine that, to mine or biomine them, and less how to keep the process sustainable, because of course we want everything to be sustainable. So more research is needed from this point of view, and but nevertheless, the BioRock experiment at least, and also the other experiment that have been performed on this topic, demonstrated that not only these processes are possible for extraterrestrial human settlement, of course, not in the near future, but in future, but also it demonstrates that it is useful on Earth. In fact, as I told you, we discovered that Pingmonal dissecabilis as a novel rare earth elements biomining activity that was not known before, and that can be successfully used on Earth as well. So with that, I would like to thank you for your attention. And I leave you with some useful links here that if you want to know more about biomining in space, but in general about our research and space microbiology in general. And thank you so much.
Well, thank you, Rosa. Perhaps people could unmute.
Thank you.
It's always difficult doing these Zoom meetings and having some personal connection. But thank you. That was a great insight into, I guess, a little known area of experimentation on the space station. Peter, questions?
Yes, we've got a couple from the members, and then I think there's a few on YouTube to be dealt with. Sean's asked a typical Sean question.
Thanks very much, Peter.
That was a compliment, right?
That was a compliment. Would you like to maybe simplify the question a bit, Sean?
Yes. Firstly, I just wanted to take the opportunity to thank Dr. Santomartino for a fantastic presentation. What a worthwhile thing to do with your life, to be involved in that kind of work, because I think it's the answer to all those skeptics who say, why should we spend money on science and research? It was really about the point you were making at the end about the distance we have to go in terms of being able to commercially mine, extract, process, and return minerals to Earth. And you alluded to some of the problems there, and I was wondering around things like, you know, exposure of microorganisms to microwave radiation in space, and how that might, you know, degrade their effectiveness, and how you extract minerals from microorganisms and practically get them to somewhere where they can be refined to the stage they can be commercially useful here on Earth, because economics drives so much of this. And I just wondered if you had any views on how long we're talking before we might practically be at that stage. Are we 50 years, 100 years?
That's one of the main question, I think, because if you think about it, the interest is quite new in the field, and thinking about the fact that BioRock is basically it's been the first biomining experiment on a space station, you can understand how far we are, because BioRock was quite, well, of course, it's my experiment, so I have to say it was very nice, but it is quite a good experiment, but it was the first experiment. And also when I showed you the hardware, it was a very well developed hardware, but was still an hardware more for research than for real biomining, because the amount of liquid culture that you can have, and also the rock material that you can have, is quite limited. For instance, we could only put a basal slide that is 1.3 per 1.3 centimeters. So imagine a very, very small amount. And we are going to put, for bioacid, we are going to use an amount that is less than 1 gram of asteroidal material. So considering that this is the first biomining experiment on a space station, you can understand how far we are to a real industrial application. As I said, there are a lot of prototypes of basically, maybe prototype is not the right word, but more ideas and projects that are trying to address this issue, how to understand that in particular, so for example, when we think about biomining of Moon and Mars, maybe we are closer, but first we have to think about how to create a human outpost there, and we are still far from understanding that. Moon is maybe is probably going to be the first one. Mars is going to be the second, and then last we are going to understand the bioasteroid. But I don't have, this is just my opinion, I don't have a precise answer to that. Order of times, I would say that is not going to be first and 30 years, maybe, because they have to, well, maybe from the point of view of extracting from Moon, it will be in the orders of a decade, if we want to be very, very positive.
Wow.
For Mars, probably something more. But for asteroids, it's even harder because they are still trying to understand how to practically doing that, because you know that there is the asteroid belt between Mars and Jupiter. And a lot of asteroids and meteorites that are present there are rich in very extremely useful and precious elements. So there is a lot of interest in biomine them. However, how are you going to do that? Are you going to collect them, capture them, and take them on Earth, or are you going to? There are some prototypes that I saw online that basically think about going there, collecting with the balloon, basically enveloping the meteorite with a balloon, and then injecting the material, or at least the leaching chemicals surrounding the asteroid, bio-leaching it, and then how are you going to collect that material and take it back to Earth? It's going to be quite expensive, I think. So they are still in the moment in which they are trying to understand how to make it costly effective and not much expensive in respect to the advantages that this takes. I don't know if I probably didn't answer your question, but it's
No,
because I didn't have an answer.
No, thank you. That was a great response. Thank you very much.
I think, is it fair to say, Rosa, that basically in the early stages of this, it's really about trying to make bases on the Moon or Mars sustainable and actually not have to carry so much stuff out
Yeah.
there. Okay, Neil, you had a question. I think some of that was answered. Do you want to elaborate?
Yeah, thanks, Peter. Yes, it was indeed. I think, first of all, just to reiterate what Sean said, fascinating story, and it's really great to hear that the science is happening on our doorstep. So yeah, I really enjoyed the presentation. You did answer the first part of my question, which was what are the typical durations of the experiments? So we got that 21 days. The second part was being able to compare the yield of any reaction, because obviously when you're waiting for the yeast to prove so you can make your pizza, you know that it's a couple of hours. But if we have to wait months for the soil to be ready when we're on the Moon, will that be a problem? So how do we work out the yield of the reactions?
So in biotechnological, and I think in general bioindustrial processes, you start with something like we did, so very small experiment in which you try to understand if the process works, first of all, and then if the process works, you go on with the scaling up of the system. This is true for every type of bioindustrial and biotechnological application, but this is particularly true in this case. So I think that now we have a limitation, that is that the International Space Station, it's a fascinating facility, but we have some limitations in the amount of material that we can send there, of course. For instance, again, sorry to repeat myself, but the biomining reactor that we developed had that size because it was necessary to be integrated into an incubator that was already present on the International Space Station. There is the Cubic Incubator. It's a European Space Agency facility. If we want to scale up the system, I think that what will be very good to do is first to try to scale up the system here on Earth. And there are several groups that are already doing that. For instance, the University of Colorado, but also at NASA, I know that people is doing that. But there is a but. That is that from the point of view of microorganisms, simulated microgravity that we can do here on Earth is not the same as real microgravity, because microorganisms are very, as I showed you before, they are very tiny. And it is believed that small microorganisms cannot actually sense microgravity directly. And what they sense is that they sense the microgravity indirectly by a kind of environment that is fixed and not very well mixed. So, well, maybe I'm expanding too much the topic, but what is important to say is that even if we try, the first step, in my opinion, would be to try develop these systems here on Earth and then try to transfer them on space, maybe first on a space facility and then maybe on Moon and on Mars, and then scaling up the system more and more, and then trying to understand at which point we have to adjust the things. Like, for example, it may be that at some points what is feasible in 5 ml, in a volume of 5 ml, this is what we had for BioRock experiment, is not enough for a bigger experiment. So you have to, I don't know, maybe give them more nutrients or add them agitation or something like that. So the scaling up of the process will be absolutely necessary if we want this process to become really useful for human settlement.
Thank you for that.
Thank you.
Geraldine and Carl have a question on biosecurity. Do you want to ask it?
Rosa, thank you very much for the talk. It was really good. I'm curious about what happens if the wee miners escape. You know, you talk about having a small volume, but what happens if a very, very small amount of it gets out?
That's a very good question. And actually, people that is thinking about the possibility of doing this kind of biotechnological any kind of biotechnology on outer space, they have to deal with this, the topic of planetary protection. So you may know that there are some laws of planetary protection. I'm not particularly an expert on that, but what I know is that, for example, on Moon, Moon should be not particularly protected from any law, while Mars is. The issue is that if one day we are going to create a human outpost on another planet, we are going to bring our bacteria with us. And if the outpost is going to be a permanent settlement, then the problem of planetary protection, I think, it won't exist anymore, because if we are going to live there freely, we are going to take our bacteria with us. So we are first of all contaminating somehow the environment. The problem exists. Yeah, I think I may say I'm not an expert on the topic, but in the frame of a space station, that's not an issue, because the bacteria that we use are completely safe. So even if they escaped from the biomining reactor, they would have been completely safe, and they would have been no harm at all for the astronaut. From the point of view of future outposts, then I think it needs to be discussed by government, probably, institutions, and we need to think of, at that point, we need to think about, okay, is this going to be a permanent settlement? Is this going to be just a settlement that is there, but we are not going to live there. So in that case, maybe we may think about creating a closed environment that doesn't contaminate the body, the planetary body that we are in, and so on.
Can we have a couple of quick questions and then move to the
Thank
YouTube
you.
questions? Will, you had a question about mining asteroids on Earth, meteorites.
Yeah. It was a marvelous talk, Dr. Rosa Santo, and absolutely brilliant. Having a big interest in mining myself and also meteorites, I just wondered if loads of tests have been done on biomining on terrestrial meteorites that have been found here, and all the different types. And I just wondered what sort of results was it looking like they were getting very good results with things? And obviously you're going to take it out to the space station to try a similar material to asteroids. And final part of the question is the famous Allen Hills meteorite, which was found, that came from Mars. And the signatures, do you sort of believe that the signature of the famous one that Bill Clinton said, life has been found on Mars, all this kind of stuff, could this be signs that biomining has already taken place on Mars? Or, if you see what I mean, or has already taken place on a meteorite which has come here from Mars, if you see what I mean.
So regarding the first question, yes, there are groups that already tested biomining of asteroids. I am sorry that I don't remember exactly the names of the groups, but I'm pretty sure that they did that in US for sure. I know that they are studying something similar, as I say, in the University of Colorado, and they are studying that at NASA. And also there are several groups that are doing that. In our laboratory, there is Annemiek, the PhD student that I showed you in the images that helped us for BioRock, that is not studying biomining in particular, but she's trying to understand if a community of microorganisms can grow on asteroids, and there are a lot of experiments on that. Of course, for bioasteroid, we are going to test this before the launch. And this is, unfortunately, the COVID situation blocked us a little bit because in the previous 3 months, this is exactly what I should have done. So try to test, to take my organism and try to test what they could mine the asteroid. But unfortunately, my experiment blocked a little bit, and now I finally came back to work and I'm preparing everything for the experiment. So yes, it is already, it's been tested on Earth and it has been successful. I know that they extracted some useful elements, in particular metals, of course, and I think, I may be wrong, but I think they extracted platinum for sure, and then a lot of no, I don't want to say something that is not correct, but I'm sure that they successfully extracted something from asteroids. And I think they also used fungus, unicellular fungus in this case. The second question was, sorry?
Yeah,
again?
I just wondered that there was the famous meteorite which was found from Mars many years ago, and sort of Bill Clinton sort of brought it out to the world saying this is perhaps signs of life on Mars, but it was taken away because people didn't recognize, sort of the little holes and stuff which could have been formed by microorganisms. There was, the scientific community couldn't be sure, so it was kind of swept under the carpet and forgotten about. But I just wondered if that was coming in with, maybe with this biomining, you might see similar effects on material that might sort of show these kind of effects, if you see what I mean.
Oh, I see. Yeah, well, in the case of that meteorite, there were several reasons why in the end, they understand that it was not a biosignature because they could explain that feature by chemical processes. So they could not demonstrate it was biological. And there is a whole bunch of a lot of studies about biosignatures and fossils on biosignatures. It's not my topic, but I know that this is what happens to this meteorite. However, it is possible that one of them, if there was life on Mars, or if so what I'm trying to say is that when microorganisms bio-leach a rock, they really weather it. And so it is possible to visualize the effect of the weathered rock at the microscope, at the electronic microscope. So yes, if this happens, I believe that maybe it will be possible to see that. However, I think we will need to be extremely sure that was made, that signature is actually a biosignature and not a chemical biosignature. And personally, I won't be able to do that because you need more the point of view of a geologist to know that. I'm a pure microbiologist.
Marvelous, thank you so much.
Thank you for that. And the last question from the members is from Sarah. Sarah, do you want to put your question or will I ask it?
Yeah, it was just about, I mean, it's probably early days, but in terms of the genetics and any changes that might occur, and if anyone's looking into that, the effects on space, like how they're adapting.
Mm-hmm. Yeah, there are a lot of studies about that. In fact, the whole branch of space microbiology, there is a study this, the effect of not only microgravity, but space conditions in general on microorganisms, of course. And there are a lot of studies that demonstrate the effect of genetics from different points of view. The majority of studies study actually not really what happens from the point of view of growth, but more bacteria or microorganisms in general were exposed to external facilities on the International Space Station for a certain time, depending on the experiment. Then but they were inactivated. Then the samples came back on Earth, and they tried to understand if there were any mutation, if they survived this capacity, so if they survived the radiation of outer space and this kind of experiment. But I know that there are also experiments performed inside the International Space Station. When I say inside the International Space Station, I mean that in that case, they not only exposed the microorganism outside in a dried deactivated state, but they were actually growing, and they saw what happens during experiment and after the experiment. And the result that they show is that there are a lot of mutations that are accumulating over the space experiment. Then there are microorganisms that are more or less resistant to these mutations. Sometimes they get to produce spores, sometimes they get to produce get more resistant to some conditions, then sometimes they discover to be not resistant at all to this to the environment, to the space conditions, so they died. It depends on the organisms, and this is really a big question in the field of space microbiology, because microbiology is so wide, microorganisms are so wide and so diverse, there is not a single truth. So I cannot tell you, yes, microorganisms resist to that, or no, they cannot resist to that, or yes, there are genetic changes, no, there are not genetic changes. It really depends on the microorganisms and on the specific species. Yeah. But yes, there are a lot of experiments on that. To my understanding, there is only one. From my knowledge, there are few experiments that studied the effect of bioleaching in space from the genetic point of view. I'm sure that there is at least one on Cubriavitus medallidurans. There is one of the 3 organisms that we sent for BioRock, because it was an experiment performed in preparation for BioRock. But yeah, there are a lot of experiments on the topic, but I think not a lot regarding biomining and genetic changes in particular.
Yeah.
Although we should study that, definitely.
Thank you. It's interesting. Thanks.
Can I hand over to Mark for the YouTube questions?
Yeah, we've had a lot of interest from our YouTube audience, so we could be here all night with these questions. Graham had a question about mutations as well. I think you've just answered that one, so we'll move on from that one. Hans is asking, is it a mix of microorganisms that contribute to all these applications, or is it one or two microorganisms that tend to be productive or useful? And I'll add to that, Graham's also asking, is it unicellular microbes for bioleaching, biomining, or is it colonies and symbiotes such as fungi and algae?
It depends on what you are observing. So as I said, when we look at the natural environment, they are 99.99% of the time it's going to be a community, because of course a natural environment, being natural, is full of different types of microorganisms, and in that case probably the community works better. Indeed, when we want to study an organisms for a bioindustrial process, then in that case we need to study if it is better to use a community or a single organisms. So for simplicity, and also in order to understand the effect of the single organisms, in this case we decided to study the single organisms, so the Singomonas dissecabilis, or Bacillus subtilis, or the third one, that is Cubriavidus metallidurans. However, for bioasteroids, we decided to use single organisms and the community together, because as they correctly asked, sometimes the community works better. Sometimes it's not always the case, because it depends on the equilibriums that is created. If you have a whole bunch of microorganisms, so a community, they normally reach an equilibrium in which they behave, I explained to you, for instance, the Melissa project. What happens normally when you have a community is that one microorganisms creates something that is useful to another microorganisms that produce something that is useful to another microorganisms, and in this way the equilibrium is created, and it's very it works much better than the single organism. However, they could also compete. So again, if we are looking to a natural environment, they will create their own equilibrium. But if we want to move this process to laboratory controlled environment, then in that case, we are not as good as nature. So we have to understand which community is the best. Is it better the single one? Is it better to use this organism with this other? Are they okay? Not okay? Maybe it's better if we use these other organisms. So this needs to be tested. And maybe another question was more regarding the real biotechnological application. And the answer to that is that it really depends on what you want to do. So I guess every biotechnological application needs to find their own organism or organisms that works better together or alone. I hope I answered.
Fine.
Shristha asks, could you cite examples where microbes have been used industrially to improve metal recovery from mine wastes?
Well, there is a very wide, the most commonly used bioremediation microorganisms is the so-called Acidobacillus peroxidans, and thioperoxidant also. This is a bacterium that has been widely studied because it has a lot of application. It is, so maybe I should start saying that when we talk about bioremediation, bioleaching, biomining, the most widely used and most studied organisms is this one. This one is an organism that lives in a very, very low pH. And thanks to this very low pH, it is able to extract useful minerals and elements from the rocks or from the waste better than the others. However, from the bioindustrial point of view, they are trying to understand if this process, if there are better microorganisms than this one, just because it requires such extreme environments, because a low pH environment is a little extreme. And of course, if you think about the process of bioremediation, the ideal bioremediation process is the one in which you just pick the microorganisms or the bunch of microorganisms that do that cleans the polluted environment and cleans it. So what you want to do is theoretically spread this collection of organisms on the polluted environment. But of course, in that case, you have to be careful if I'm not sure how often this are used, because they have to be careful that they are not spraying something that, yes, maybe cleans the environment, but then it breaks some equilibrium and you cannot restore the equilibrium back. So there are bioethics and also a lot of biotechnological laws and issues that need to be addressed when you want to do something like that. Although it's widely used.
Okay, have a couple more. What living factors will you use to produce soil, considering that the soil on Earth is composed from living and non-living elements?
That's a good question that I don't know how expert I am in soil, because you need to be more expert in soil science. There's a whole bunch of science to do that. So I think maybe the safer answer would be that I will need to study that and then decide. But of course, what you need is the right type of microorganisms. So as I said before, you first have to select the one that are the best ones. Probably I will use a community, and you will need to the nutrients that are necessary for these microorganisms in order to flourish and to do their tasks freely and without the intervention of humans as much as possible, so that the process is more natural as possible,
I'll ask one more, we could keep going. Someone's asking, how is bioleaching different from bioming?
They are sometimes referred as synonyms. I like to distinguish the two because I refer to bioleaching more to the biochemical processes, while biomining is more really the industrial application of bioleaching, sorry. But they are used as synonyms very, very often. So it's not wrong to say that use them as synonyms. I just prefer, for my brain clarity, that when I talk about bioleaching, I talk about the process, and when I talk about biomining, I'm talking about the industrial process that takes advantage of the bioleaching experiment and processes.
And one from me, have you applied for the astronaut program so you can do this yourself on the space station?
No, because I don't want to be an astronaut, but my partner wants to. So it's really, so I'm doing the research, and maybe I'm going to give him the samples that is going to. No, first of all, I have contact lenses, but I am too blind to be an astronaut. Also, no, it's too much for me. I like to. It was very, very extremely intense to have from a positive point of view, get the samples back, because you get something that was in space. And also when you were preparing the samples, I was feeling like they were my children. Oh, my children are going to go to space. And then when they took the pictures of the astronauts, Luca Parmitano, that was working on the samples, I was looking at this picture and I was like, that hardware, I actually prepared that hardware. And when it came back, I thought that they were in space. So I like this kind of feeling, but I don't think I would be good enough to be an astronaut.
It's passion that makes science interesting.
Yeah.
That's what we want. Can I just ask everybody to say thank you very much? That was a fantastic talk. Thank you very much.
Thank you very much. Thank you for inviting me.
And you were kind enough to put your email address on the slide.
Yes.
So I guess if somebody desperately wanted to ask another question,
Yeah.
you may get an email. But thank you very much.
Thank you so much for inviting me. It was very nice to be here.
Pleasure. Thank you. Just a reminder that next Wednesday it's the imaging group. It's open to all ASE members, but it is a members only meeting. But I look forward to seeing you all there. Thank you very much. Good night.
Good night.
Good night. Thank you.
Good night.